Good morning, Vanchika. So, I am Rahul Saha, and today we'll be finishing up that worksheet from the molecular basis of inheritance and proceeding with the next worksheet that I had sent it to you on your group regarding the principles of inheritance. Okay. So we had completed till question number 131. So come to the older worksheet, question number 132. Let's finish that off. So let me open it. Okay, so question number 132. Yeah. During DNA replication, the strands separate by. That's a very easy question. The strands separate means the denaturation of the strands. Okay, the DNA remodeling happens by DNA topoisomerase, polymerase, unwindase, helicase, or gyrase. So the answer to this question is simply, you can say, we have a specific enzyme known as DNA helicase, right? That is the DNA B in case of prokaryotes. So when it comes to DNA helicase, it is an ATPase molecule, uses ATP hydrolysis, right, to derive energy. First characteristic is this one. So these questions can be asked. Next thing is it's a hexamer. It's made up of 6 polypeptides. Okay, sequences comes in with 6 monomeric helicase loaders. Helicase loaders, right? So we have the DNA B, DNA C complex in case of prokaryotes. Okay, same way in case of eukaryotes. Okay, it is known by the name, we can say MCM, mini chromosome maintenance complex. So that can be asked sometimes. Okay, so I'll write it down for you. So in case of eukaryotes, it is known as MCM. Okay, same helicase. So in case of prokaryotes, it is known as DNA B. Okay, helicase loader is DNA C. Okay, in case of prokaryotes, this is the thing. In case of eukaryotes, it is known as MCM, mini chromosome maintenance complex. From 2 to 7, you can count 2, 3, 4, 5, 6, 7. You have 6 polypeptide subunits. Okay, 3 are catalytic, 3 are regulatory. Okay, so that is not in your course. Just know MCM. Okay, that is enough. Then coming on to the next question. And one more thing, when it has done the denaturation, who will bind SSB, single strand binding proteins? Okay, to keep that thing or keep those strands which have been denatured in the denatured state only. Okay, so now second thing, question number 133. If you come to, again a very easy question. The process of translation is directly protein synthesis. Nothing else happens. Translation means in the center dogma mRNA to protein synthesis, right? Option number B. Now question number 134. A DNA with unequal nitrogen bases would most probably be, just think, unequal nitrogen bases means it is not following the Chargaff's law. So in one of those questions we had talked about that single stranded molecules like RNA do not follow Chargaff's law, right? Neither first parity nor the second parity law. We have already discussed this in detail in our previous classes. Chargaff's law, no need to repeat it. We have the first parity law, double strand. In single strand also, number of A's are equal to number of T's, number of C's are equal to number of G's. Okay, in even the single strand, that is the second parity law. First parity and second parity is demonstrated by the thermodynamic stability. Okay. So now what we have is we have something known as Chargaff's law of first parity and second parity, but that is not followed by intrinsically single-stranded molecules, SS molecules like RNA. Okay, so the answer will be single-stranded for 134. 135. Nucleotide arrangement in DNA can be seen by electron microscope, ultracentrifuge, light microscope. Light microscope without no use you cannot reach DNA. Ultracentrifugation will go for, you can say, differentiation as per the density useless. Electron microscope, you need to use a 24 karat gold coin and impregnate DNA. You can see the structure of the cell in the nano angstrom structure, but arrangement you can never see. Arrangement, you have to crystallize DNA. First we need to crystallize DNA and then we bombard DNA with X-rays and check the elastic plus inelastic collision. Mostly the collisions are inelastic only. There is no real elastic collision. Ideal collision is elastic. So we check the diffracted rays of X-ray. Okay, and then from that we deduce the structure of DNA as it was deduced by Rosalind Franklin, then Maurice Wilkins, then republished by James T. Watson and Francis Crick, right? So nowadays there is a new technique. Just for your knowledge, I am telling X-ray crystallography is the correct answer, because what you do is you make crystals and then you bombard them with X-ray and then you get the structure. Okay, the structure of the proteins. You might have seen many, you can say, quaternary structure proteins that I have shown you. Okay, those folded structures, those are all deduced by X-ray crystallography. But nowadays there is a new technique known as cryo-electron microscopy. Okay, this got Nobel Prize in 2013 or 14. So in chemistry it got Nobel Prize, but the technique is used heavily in biology. In minus 196 degree Celsius liquid nitrogen. Now it has reached the atomic resolution barrier also. In the last month there was a research paper that it showed that by cryo-electron microscopy it is much easier than X-ray crystallography, because sometimes many proteins you cannot form crystals. Right? You cannot form crystals in case of multiple proteins, but that is not the issue with cryo-electron microscopy. So nowadays we use this, but as per the option, this is correct. Okay, X-ray crystallography. Then experimental material. Question number 136. Experimental material in the study of DNA replication has been. So I have taught you multiple times that the model organism is gram-negative microorganism, the bacterium Escherichia coli. Right? Escherichia coli. Okay. 6 million base pairs. Okay, one chromosome only. Okay. Remains haploid. Entire DNA is one chromosome only. Okay. 20 minutes is the generation time, and it is, you can say, as per the gram stain, the old staining technique is gram negative in nature. Okay. So the thing is, coming on to question number 137. Okay. Khorana first deciphered the triplet codons. Okay. So we all know about, you can say, the genetic code. So his complete name was Hargobind Khorana. He was a biochemist, right? And he was awarded the Nobel Prize, also the most prestigious Nobel Prize as well. Okay. So we know that he was the one who deciphered the triplet codons. Okay. He was the one who really deciphered the triplet codons. Hargobind Khorana. But the thing is that this question says Khorana first deciphered the triplet codon of. Okay. So what was the first two in case of Hargobind Khorana? Okay. So the thing is, here if we talk about Question number 137. So look at the options: serine and isoleucine, cysteine and valine. Okay. Then we have tyrosine and tryptophan. Okay. And then we have phenylalanine and methionine. So we have multiple options. So the correct answer is cysteine and valine. These were the first two amino acids which were deciphered, or the genetic code of these were deciphered by Hargobind Khurana, cysteine and valine. Okay, so you got to remember this because this was an experimental question. Okay, so in many cases, in many research papers, it is also counteracted that he discovered the first triplet codon of threonine and histidine also. But as per NCERT, go with cysteine and valine. Okay, I'm just telling you the alternative facts also, but go with cysteine and valine as per this. Now, Escherichia coli come to Matt Michelson and Franklin Stahl experiment, Michelson Stahl. So, Escherichia coli fully labeled with 15N is allowed to grow in 14N medium fine. Now, the two strands of DNA molecule of the first generation of bacteria have option number A, different density and do not resemble parent DNA. B is different density but resembles parent DNA. Same density and resemble parent DNA, same density but do not resemble parent DNA. So same density would never be there because you are changing the isotope. From 15N heavy you are going to 14N light. That's why we went for cesium chloride density gradient centrifugation so that it can detect this minute 1 atomic mass unit change also in the atomic mass. So last two options are incorrect. Coming on to A and B, different density but resemble parent DNA. How will you resemble parent DNA if you have different density? Because let's draw a small graph. You have 15N bands, so all will be 15N here. Okay, what you have is you have a 14N band, suppose all will be here. So after this is generation 0. I had explained this to you when I was teaching you Matt Michelson and Franklin Stahl experiment. A semi-conservative method of DNA replication was uncovered by these two scientists. So now generation 0 is this one, generation first. So these will act as template, but since only 14N are present in the medium, new strand will be 14N. So they will be having a density intermediate between 14N and 15N in generation 1. Okay. So one of the strands will be 15N parental. Okay. Template, whereas this daughter strand will be your 14N. So the thing is they will be somewhere here, but now second generation wise this can act as template. So this will become thinner, this will become thinner, and you will have a complete band here from generation 2 onwards. Okay, because this is acting as a template, so you will be having both, you can say 3 to 5 prime, 5 to 3 prime as your DNA daughter strand. This is daughter, okay, this is parental, but still this is 14N. Okay, so the thing is, after generation 1 also the densities will not be same. It will be intermediate density. So I had explained to you that after generation 1 you would be ruling out the completely conservative method. If completely conservative method is present, then this midway would not have occurred. Okay, directly you would have formed 14N, and then this can rule out completely conservative, but dispersive is still there. Now dispersive is ruled out in generation 2, right? So even in generation 1 the densities will never be the same. Okay, the densities will never be the same. Good morning Ayushi, good morning Ayman, good evening, good morning Tarun. Okay, so the thing is density will never be the same. So for question number 1, you are in with 138, the answer will be 1. Different density and do not resemble parent DNA. Is it resembling parent DNA? Parent DNA are both 15N, 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 both. Okay, It is not resembling. It is 115N, 114N. Okay, this 114N that I have written already. Not resembling parent DNA, not it is not having the same density again. That is the reasoning behind this question. Coming on to question number 139. Okay. The process of transfer of genetic information from DNA to RNA. Formation of RNA from DNA is transcription. Simple as that, and option number B. Now, question number 140. An octamer of histone complexed with DNA forms a nucleosome. Multiple times we have talked about nucleosome. Okay, that is an octamer of histone, H3 2, H4 2, whereas H2A, H2B dimers hold twice. Okay, so it is 8 histones. Okay, so 8 histones. You can say polypeptide chains, but there are 4 core histones here. So that is the meaning of this question. 4 core histones. They have not written core histone. So we need to decipher that they are talking about core histones only. And then you will be forming this thing. Histone octamer, and DNA will be wrapped around it. Okay, 1.67 times, 6567 times in a left-handed superhelical way. Okay, 146 to 148 nucleotides of DNA are wrapped around it. Okay, this is known as the nucleosome. Then we will be having the chromatosome, microfocal, okay, nuclease. So we have talked about all that in the last class itself. Okay, now coming on to the next question. In the genetic dictionary, there are 64 codons as. Coming on to question number 141. Okay, 64 amino acids are to be coded? No, because 61 are amino acids, 3 are the stop codons, right? UAA, UGA and UAG, right? Now 64 times tRNA present? No, there are 44 nonsense codons. That is another, you can say, nonsense complete and nonsense statement, and 20 sense codons. The only correct option is genetic code is triplet. Okay, the significance I had given to you, answer number, option number D, 4 to the power 1, only 4, 4 to the power 4, 4 into 4, 16. Now only option is 4 to the power 3, 4 into 4 into 4, 64, because you have at least 20 essential amino acids. So if you keep the genetic code doublet, you cannot have. Triplet will answer it, and thereby you will generate degeneracy also, where you can say multiple codons can code for one amino acid, but you have ambiguity also. Genetic code is ambiguous because one codon cannot code for more than one amino acid. It only codes for more than one amino. It only codes for one amino acid. Okay, and methionine and tryptophan, they do not show the degeneracy. Only 2 which are coded by only one codon each, whereas serine shows maximum degeneracy. It is coded by 6 different codons. Okay, and 3 codons, 61 are, you can say, sense codons in that sense, and 3 will be your completely nonsense ones, stop codon. Okay, 3 are UA. Okay, so this is the correct answer. Coming on to the next question. DNA replication is, very good question. This is 142. Conservative and discontinuous. Conservative, A and D are wrong. Come to B and C. Semi-conservative and semi-discontinuous. Semi-conservative and discontinuous. What will be the answer? Very good question. Question number 142. What will be the answer? Just think about it. Just try answering it without looking at the answer sheet. Try answering it. We have talked about it in detail when I was teaching you this in class. I had given you both the models of simple Okazaki fragment model and the currently accepted model, which is known as the trombone loop model, that I had also given it to you for your complete understanding. The thing is, it is known as semi-discontinuous because one strand is continuous, the leading strand. The 3 to 5 prime parental template will go for direct. You will be having an RNA primer 5 to 3 prime. OH, and then you can synthesize easygoing, okay, via DNA polymerase 3, okay, which is a holoenzyme, multimeric holoenzyme. DNA pol 3, clamp loader will come, clamp beta clamp will come, right? And that will lead to synthesis of specific leading strand. This is the leading strand, continuous. But the second template strand will be 5' to 3'. Then we will be coming on to the replication fork model here, because two replication forks are opened at one time, right? Bidirectional replication. And then what happens is, when you open it up, this is the 5' to 3'. So you cannot go for synthesis like this because this is not opened and you do not have enough energy, okay, to open it, because if you spend so much energy in opening, you need to again go for SSBs, and SSBs may not be present in, you can say, that much amount at one go. It may renature again, okay. So due to the energy constraint, entire thing is not, you can say, unwounded, and then superhelical you can say twists will also occur, right? Those are known as the torsional strain, okay, supercoiling. So that will be again needed to be resolved by other topoisomerase class 1 or class 2 using ATP or non-ATP, using single stranded or non-stranded DNA or double stranded DNA as the template directly. So here what happens is, a 5' to 3' primer is produced. This is the model given in your NCERT book, and then this again it will backtrack 5' to 3' primer, RNA primer. Again this one, okay. So here is the 3 to 5' synthesis cannot happen via DNA ligase, okay. 5 to 3 can happen, 3 to 5 cannot happen from this direction, okay. Then by DNA polymerase 1, sorry. So what happens is, for this specific mode of phosphodiester bond formation, you need DNA ligase in the end, okay. Otherwise DNA polymerases also have, you can say, the ligase activity, because they keep on synthesizing their DNA, right? So without forming phosphodiester bond, how come they are synthesizing? The kind of ligase activity you can say, but from 5 to 3 direction, not from 3 to 5, okay. So it is 3 to 5', so DNA ligase is needed for this specific thing, because I had explained to you that this is a very intricate detail, because you may think that polymerase can only do it, but polymerase is at the 3' facing the 5', it cannot do it. 5 to 3 it can do, right? Just the opposite. But here it cannot do it because it will be facing the 3' here, okay, and 5' is here, 3' OH will be here. So it cannot do it the opposite way along, okay, because 5' is phosphate is here, 3' OH will be here, so it cannot do it, okay. When 5' phosphate is there to 3' OH, then it can, you can say, directly it can do from opposite orientation, okay. So thereby it is a very good combination, from starting from 3' OH, okay, it can directly generate, okay, 3' OH to a new one it can generate, but it cannot generate the other way around. That is the thing for DNA polymerase, okay, that can be done by DNA ligases, okay. So the thing is, this is known as discontinuous, but half was, this one was completely continuous, so the entire thing is semi-discontinuous. Semi-discontinuous mode of replication. That is true, and it is semi-conservative, so that is also true. So semi-conservative and semi-discontinuous mode of replication is correct. So next we'll come on to the next question. Genetic code consists of, question number 143. All adenine, guanine, cytosine, uracil, okay, all will be present in the genetic code, Okay, that is the correct answer is option number D. Okay, so now we have finished this worksheet regarding the molecular basis of inheritance. Okay, so let's close this worksheet. Now you can open your next worksheet, right, which concerns the chapter number 5, which has more than 140 questions again. Principles of inheritance and variation. Okay, the genetics chapter that I used likely call it. That is the molecular biology. This is the core genetics. Okay, not quantitative. We have only qualitative genetics in your course and little bit of combining with linkage and recombination modern genetics, but mostly you deal with classical genetics proposed by Gregor Johann Mendel. So come to question number 1, a very good question. Thalassemia and sickle cell anemia are caused due to a problem in globin molecule synthesis. Okay, select the correct statement. Now A is both are due to a quantitative defect. Just understand this specific thing. Just don't go for that is the correct answer. Fine. In order to know the correct answer, you should know also why the other options are incorrect. That gives you the perfect knowledge. Okay. There are people who will explain to you why that is the correct answer, but there will be very few people who will explain to you why the other 3 options are incorrect. Okay. So understand it completely for your thorough knowledge so that you can not be trapped in the question if it is asked in any other way. Option A, both are due to a quantitative defect in globin chain synthesis. B, thalassemia is due to less synthesis of globin molecule. Okay. Sickle cell anemia is due to a quantitative problem of globin molecules. Both are due to a qualitative defect in globin chain synthesis. Okay. The answer to this question is B. Thalassemia is due to less synthesis of globin molecules. That is the correct statement. But why the 3 are incorrect? What happens is, when we talk about thalassemia, we can have alpha thalassemia, beta thalassemia, because adult hemoglobin is comprised of this one. Okay, whereas fetal has this one. Okay, then you can have in embryonic hemoglobin also, this is fetal. Okay, always know this comes also in NEET examination. For information, this is fetal hemoglobin, this is adult hemoglobin. Okay, in embryo you have zeta 2 epsilon 2, that is known as Gower 1, and alpha 2 epsilon 2, that is known as Gower 2. This is in embryo. So know these 4 types. These genes are also present in adult. This one, zeta, epsilon and gamma. The genes are never deleted. You will think that how come genes got deleted. What is the answer? That the beta gene is epigenetically repressed, epigenetically silenced, too much heavily methylated and non-acetylation. Whereas when we switch from embryo to fetal stage, the zeta and epsilon, they get epigenetically silenced for life. They enter the, you can say, constitutive heterochromatin region. Okay, that is, you can say it is formed on top of those genes so that RNA polymerase 2 can never get access to the upstream promoter and enhancer sequences in the eukaryotic genome present upstream of these specific genes. And when the fetus is delivered, so after a week or so, all the gamma, you can say, gene coding the gamma polypeptide, the gamma gene is also silenced epigenetically, and beta is epigenetically non-silenced or unsilenced. And then we have alpha 2, beta 2. So gamma doesn't bind to, there is a reason also in NEET examination if you consider class 11th respiration. Okay, so respiratory system, if you have read properly in 11th, you will be knowing that there is a graph of hemoglobin saturation. Okay, oxygen saturation of hemoglobin, okay, oxygen content, and this side is HbO2. So the thing is, that graph will be not affected by 2,3-bisphosphoglycerate, okay, which is formed in also high altitude conditions. So the beta chain binds to 2,3-BPG, and binding upon that, the HbO2 unloads O2 and becomes deoxygenated hemoglobin. But if in case of fetal hemoglobin, this has no effect because there is no gamma. Because fetus doesn't take air, it is dependent upon mother's blood for oxygen, it cannot afford that. If mother's blood has too much through 3 bpg, it will become you can say deoxygenated hemoglobin. So it cannot afford that, because if it becomes, it cannot go for air that it will get oxygen. It has to wait until the metabolites like 2-3 bpg are cleared, then it will die also. That is the evolutionary relationship and the real reason why in fetus gamma chain is present. Okay, so everything in biology has a definitive reason. Now thalassemia means the alpha chain and beta chain are synthesized. They have a specific mutation. They are synthesized in very, very less quantity. Okay, very important this thing that you should be knowing this. Very, very less quantity means it's a quantitative defect. Quantitative defect, because I told you, explained to you that in case of codominance, what was happening in case of this thing, snapdragon? Why was it becoming pinkish? Because you do not have enough enzyme of capital R that will convert a substance X to completely red color. Okay, so because half of the time small r is transcribed, it is not making that enzyme, so the component X, which is white in color, white pigment remains as it is, and then the blending of phenotypes, it is seen. If it's white plus red, if you mix, you will get somewhere around pink. So that is what you get. So enough synthesis is very, very important. Okay, that is why heterozygous conditions can sometimes result in, you can say, midway kind of phenotype. So it's a quantitative defect. Whereas your sickle cell anemia, both are autosomal recessive diseases. Sickle cell anemia, this is a qualitative defect, not quantitative. Okay, what happens here, the mutation, we know that in the beta chain 6th position, the glutamic acid has been changed to valine. So glutamic acid has been changed to valine. Okay. So now what happens is, this is a qualitative defect. The quantity, this remains the same, but the thing is, due to the valine, which is a neutral amino acid, again remember, this is an acidic amino acid. Because of this thing, this changes. If it was replaced by aspartic acid, okay, maybe its conformation may not have been that disconfigurational or that unstable, because aspartic acid or aspartate is an acidic amino acid, but valine is a completely neutral amino acid. Therefore, its conformational change occurs drastically and it becomes very, very sickle type. So the entire hemoglobin molecule, it assumes, it takes a sickled structure. Thereby the entire, you can say, RBC or erythrocyte, mature erythrocyte becomes sickle in shape. So this is a qualitative defect, whereas thalassemia is a quantitative defect. Now check the options. Both are quantitative, wrong. One is qualitative, one is quantitative. Sickle cell anemia is due to quantitative, wrong. It is a qualitative defect. C is wrong. D, both are qualitative, again wrong, because here again qualitative is sickle cell, but quantitative is, you can say, thalassemia. So the only correct option is thalassemia is due to less synthesis. You can see a less synthesis is quantitative, less synthesis of globin molecules. Both you can have alpha thalassemia and beta thalassemia, both. Okay. Now the genotypes of a husband and wife are, husband is IA-IB, okay. Then wife is IA-IO or IA-I, so I'll write it IA-IO for better understanding. IA-IO. So that is small I only. Now how many different genotypes or phenotypes are possible in their children? So let's do the crossing only. What does it mean IA-IB? Means IA gene will code for the enzyme N-acetyl galactosamine transferase, which will attach N-acetyl galactosamine to the last galactose. This will attach galactose to the last galactose present in the on the H substance when I taught you the H substance, if you remember, in the genetics chapter only. So that is IAIB, and here both these genes are completely silenced or mutated, you can say. So it is not making, it is only making a complete H substance only. Complete H substance means you should have that fucose also with it, fut1. Otherwise, you will go for Bombay, you can say phenotype, Bombay phenotype. Now, the thing is, let's go for crossing. Okay, when we go for crossing, IAIB, this side is your IAI. IAIA, IAIB, then IAIO, then IBIO. How many genotypes can you see? 1, 2, 3, 4. So 4 genotypes. Phenotypically is blood group A, blood group AB, blood group A again, blood group B. A, B and B, there is no O. So 3 phenotypes is the correct answer. Okay. So question number 2. Okay. So that is 4 genotypes and 3 phenotypes. Now coming on to the question number 3. Okay. A disease caused by an autosomal primary non-disjunction is autosomal primary, it is saying. So Klinefelter syndrome, okay, Turner syndrome, let's check it out. Klinefelter is XXY, Turner is X0. They are not autosomal, they are allosomal. These are all aneuploidic, but allosomal related to sex chromosomes. Okay. Next, sickle cell anemia. It is not an aneuploidic condition. It is a point mutation. So sickle cell anemia will not fall into this category. It is a Mendelian disorder, not a chromosomal disorder. Sickle cell anemia. Okay. Happens due to point mutation. It doesn't fall in this category only. So again, thinking of why the other options are wrong. Now coming to Down syndrome, trisomy on the 21st chromosome. 21st chromosome, this one, one more. So I had explained to you already that in the Pachytene stage, the crossing over will happen, the synaptonemal complex is formed in the zygotine stage, and then the dissolution of the synaptonemal complex happens in the late diplotin stage completely. But if that dissolution doesn't happen completely, if there is non-disjunction of the synaptonemal complex of or chromosomes in the meiotic event, in the meiosis cell division, then what will happen is one of the cells for one of the homologous chromosome pairing can get a triploid and one will stay like this. Okay, so that results into aneuploidy, means violation of euploidy. You are having one extra chromosome or one less chromosome in the other cell. So it happens in the 21st chromosome, that is known as Down syndrome. Okay. Then you have Patau syndrome also, that comes in your NEET examination. 13th chromosome, trisomy, same thing. Then Edwards syndrome, that is your 18th chromosome, trisomy. Okay. So the answer is Down syndrome here. Very, very easy. Now coming on to the next question. Among the following characters, which was not considered by Mendel while doing his experiments on P? Okay. So question number 4. I think it is trichomes, glandular or non-glandular. Now let's see why. So what all characteristics were considered by Mr. You can say Gregor Johann Mendel while going for his specific experiments? That is to be considered here. Okay. So when he went for his experiment, what he did was he chose 7 characteristics. Okay. So 7 characteristics Pisum sativum was chosen. Okay. 7 characteristics were chosen. So one question could be also that why Pisum sativum was chosen, okay, by Gregor Johann Mendel. Why pea plant was chosen by Mendel, you can say. So why it was chosen? Because it was easy to grow, easy to maintain. It was like it could be bred rapidly, okay, and it had proper observable discernible characteristics, okay, didn't require any specific nutrient or soil type to be grown, right? So this is why pea plant or Pisum sativum was chosen by Gregor Johann Mendel, and what all things were chosen in it? First character, flower color. You should know this completely, right? Then flower position. Then you can say stem length. Then you can say the seed shape. Then you can say seed color. Then comes pot shape. Then comes pot color. Now dominant was purple, dominant and recessive if I write. So here dominant was purple, recessive was for the color white. Second one was axial, dominant, okay, terminal, recessive. This table, just learn it properly, okay. Then tall, dwarf. Then sheet shape, round and wrinkled, that we have used already. Tall and dwarf also we had used in the monohybrid crosses. Okay, dihybrid crosses we use mainly shape and color. So the color would be yellow and green, right? 3 you would be remembering easily, but the rest 4 you should also know. Then pot shape, inflated or constricted. Constricted, okay. Then green or yellow. So this is these are the 7 characteristics that were chosen, okay, by Gregor Johann Mendel. So you just see which is not present here. Pod inflated, stem tall dwarf, it is present, okay. But trichomes, glandular, non-glandular, that is not present because those are trichomes, right? So easy question, that is. Question number 5. Which one from those given below is the period for Mendel's hybridization experiment? Question number 5. This you need to only mug it up because it's historical question. Okay, option is D, 1856 to 1863. Coming on to question number 6. The mechanism that causes a gene to move from one linkage group to another is known as, that is known as, not inversion, duplication, crossing over, okay, those are not the things. Here that will be known as, specifically you can say translocation. So it will not be inversion, okay, because what happens is we have 2 types of inversion, okay. We have paracentric, we have pericentric, okay. So what happens is, in case of paracentric inversion, what do you see? So paracentric inversion means it results when 2 breaks, okay, in one chromosome arm rejoin after excised piece has been inverted, okay. So these are mainly seen in the polytene chromosomes easily, okay, that is there. But it is not, you can say, going for any kind of rearrangement between the 2 chromosomes. So paracentric does not involve the centromere, pericentric involves the centromere, that is fine. And then when we come on to, you can say, the next option, why it is wrong. Next option is duplication. Duplication is again, gene duplication events happen because of, you can say, suppose this kind of event is happening, or because of this you are skipping it, or this kind of event is happening, so you are going for double, you can say, mode. Okay, that is called as gene duplication. In case of replication, you may form loops that will lead to duplication. Okay, that is again not exchange between the two chromosomes, right? Now, the third one is translocation. It is correct. Crossing over. Okay, that is completely a different event happening in packaging only. Okay, so it is not directly, you can say, you cannot call it translocation because it's a part of meiosis. Okay, so now translocation is just like that, it will happen. Okay, so you have reciprocal, non-reciprocal, Robertsonian. So one of the best reciprocal translocations, the example I had given to you is Philadelphia chromosome. So chromosome number 9, long arm, and again you can say chromosome number 22 is long arm. This one. This is 9. This is 22. Q arm of this one. Q arm of this one is P, petite arm. Okay, 22. So 22 already has, you can say, a simple region known as breakpoint cluster region. Okay, BCR region it has. The ABL is transferred, so some part of this one will go here, and this is the ABL region, which is the tyrosine kinase. It gets transferred here. Now this breakpoint cluster region, when it acquires ABL, it acts as a very, very strong activator of the ABL gene, tyrosine kinase gene here, and that will lead to the escape of the cell cycle checkpoints and lead to rapid multiplication of the cells. Okay, and which will lead to chronic myeloid leukemia, CML. Chronic myeloid leukemia, okay, CML disease. Okay, so translocation is the correct option. The other three are completely wrong. Coming on to question number 7. Okay, question number 7 is, if a color-blind man marries a woman who is homozygous for normal color vision, the probability of the son being color-blind is, color-blind man will have the genotype, suppose I am saying XCY, color-blind, C for color-blind. Homozygous normal means XX, go for crossing, XX, then XCY, you will get XCX, XCX carriers, then XY, okay, XY. The son can never be born colorblind if the father is colorblind, and if he is marrying a, you can say, female who is homozygous for normal vision. Okay, so the probability will be complete 0 here. Carriers will be produced in case of females, but son will be completely normal. If and only if he is not heterozygous. If he is heterozygous, if that is XC, then it could happen here XC. But here it is said that it is homozygous for normal color vision. Means both the X are completely normal. Both the X chromosomes contain the proper gene, the unmutated gene coding for the, you can say, the specific proteins, right, responsible for color blindness. So that will be 0. Okay, so you should know that it's a sex-linked, you can say, recessive trait. That much you should be knowing. Otherwise, if you can think of it as autosomal, you will be making a blunder here. Okay, in case of question number 7. Now question number 8. A cell at telophase stage is observed by a student in a plant brought from the field. He tells his teacher that this cell is not like other cells at telophase stage. There is no formation of cell plate, and thus this cell is containing more number of chromosomes as compared to other dividing cells. This would result in, very easy, you have in plants, it's not aneuploidy, Okay, it's not soma clonal variation, because soma clonal variation and polyteny, if you see, okay, they are completely different. So soma clones are completely different, you can say, soma clonal variation. So they are defined as what? Genetic variation observed among progeny of plants regenerated from somatic cells cultured in vivo. Okay, so although they must be clones, but sometimes some variations can happen. That is soma clonal variation. Okay, so that is not the case. Again, we'll go for polyteny. Okay, so polyteny is what? Polyteny is the condition of a chromosome in which DNA has repeatedly replicated without separating. That is known as endoreplication, endomitosis, like polytene chromosome. It is not happening here. There are more number of chromosomes. Separation is there. They are not saying that it has not separated. Okay, that is wrong. Aneuploidy, that is wrong. It is not one chromosome extra. Okay, more number of chromosomes compared to other dividing cells. Not more and more number, not one or two. Must be polyploidy. And since it is plants, polyploidy means a complete set of chromosome can be increased in plant cells. Okay, so since it is plants, it will not be aneuploidy. When more number it is written, not one or two, and we know that in plants complete set increases, so the answer will be polyploidy here. Okay, answer will be polyploidy. Means the entire set gets doubled or tripled, like that. So if you have 2n, it could become 2n plus n, entire set is becoming added. So you can get 3n like this. Okay. Now pick out the correct statement. Question number 9. Question number 9. Hemophilia is sex-linked recessive disease. It is a wrong statement. Okay, we have just encountered hemophilia. It's an autosomal recessive disease. Phenylketonuria is an autosomal recessive gene disorder. That is correct. Okay. Sickle cell anemia. Okay. Question number 9 I am talking about. Sorry, hemophilia is a sex-linked recessive disease. Sickle cell anemia is not a sex-linked recessive disease. Okay. So sickle cell anemia is an autosomal recessive disorder. That is wrong. Okay. Phenylketonuria is an autosomal recessive gene disorder. That is correct due to phenylalanine hydroxylase deficiency that cannot convert phenylalanine into tyrosine, right? That hydroxylation reaction cannot happen. Okay. Down syndrome is aneuploidy, it is correct. Aneuploidy means one more chromosome or one less chromosome, difference in chromosome number. So it will make chromosome number 47 from 46. Hemophilia, okay, is a sex-linked recessive disease, correct, right? It was seen first in Queen Victoria. We have hemophilia A, B, C, right? So it is deficiency of mostly clotting factor 8. Okay. So the thing is, when we talk about hemophilia A, okay, so that is the most important thing, deficiency of clotting factor 8 that we talk about. Okay. So option number 1 is correct, 2 is correct, and you can say 3 is correct. 4 is wrong because sickle cell anemia, we have just talked about the beta chain, the beta globin 1, it is a autosomal recessive disorder. So 1, 2 and 3 are correct. So that will be your option number B. That is 1, 2 and 3 are correct. Okay. Now coming to question number 10. 10 is again an easy question. Okay. A true breeding garden pea plant is crossed with a tall true breeding garden pea plant crossed with a, you can say, dwarf true breeding garden pea plant. When the F1 plants were selved, okay, the resulting genotypes were in the ratio. It is talking about genotype, okay, not phenotype. So F0 generation, capital T into small t, what you get in F1 is all will be t small t, that is done selfing. Okay, when you go for selfing, okay, T, so what you get is this thing. So talking about not the phenotypic ratio will be 3:1. 3 are tall, only 1 is dwarf. But genotypic ratio is the repetition of the allelic combinations. That is the way to know. So capital T has been repeated only once, capital T small t has been repeated twice, small t has been repeated once regardless of their phenotype, that is the genotypic ratio, so 1 to 1. Okay, so that is your, one will be tall homozygous. Okay, you can see again they have tried to make it more complicated. So one is what? Tall homozygous, not tall heterozygous. Option number 3. Option number C you can say. Then 2 is tall heterozygous, capital T small t, correct. 1 is dwarf, small t small t. So option number C will be completely true here. Okay. So I hope you are able to comprehend the lecture today. If you have any doubts, you can text me on my number. Okay. And we will be solving this worksheet in the upcoming week also, finishing 20 to 25 questions per week. So good morning, Priyanshi. Okay. Okay then. So thanks a lot. Let's meet you